So it's, as I said, VPH Summer School is multifactorial, highly multifactorial disorders. So I think the best talk we could have to start is what Ricard Solé will talk about is complexity, because highly multifactorial disorders, so you know, mathematically complexity is defined as soon as we have more than two bodies or phenomena interacting among each other, and then we cannot anymore make a deterministic prediction of what will happen. And there are a lot of theories behind this to solve this problem and to be able to tackle complexity and use complexity to develop many predictions and apply it to the real world. So maybe Ricard will say no, that I'm completely wrong in my definition of complexity. And so Ricard is researcher of the Catalan Institute for Research and Advanced Studies. So he's currently a professor at Pompeu Fabra University, and he's heading, he's reading the lab of complex systems at the PRBB, where we have the life science and department and faculty of medicine. He's also teaching in biomathematics, biological design, and complex diseases. And he has a degree in biology and in physics. So actually, he's the very representation of the integration of competencies I was mentioning before. So Ricard, thank you very much for being here. And now the word is yours.
Push here? All right. Well, thanks a lot to the organizers for inviting me. It's a huge responsibility, as I can see. So the reason probably I'm here is that I'm supposed to be an expert on complex systems, and over the last more than 10 years, we've been teaching a course here at Pompeu Fabra on complex diseases, right? And I would like to, this is a 3-month course, so I'm not going to compress everything in just an hour. But I would like to give you a glimpse of what is the philosophy behind, why it's important to think in terms of complex systems when you approach biomedical problems, and why simple mathematical models can actually be extremely powerful to answer relevant questions, right? So most of you are involved in this because you are approaching this under the computer modeling arena, where you take a realistic approach to the real complexity. And what I'm trying to sell you is that that's perfectly right, of course, but that sometimes it's good to have a very well-posed question and think in terms of the most simple approximation, because that sometimes gives you a lot of insight. So complex diseases, why complex diseases and not simple diseases? Well, it's again difficult to kind of compress the historical development of medicine, but in a way, we are here as a very successful species, unfortunately for the planet, but we are here because we have been developing a great technology. Medicine is a part of it. And over the last 100 years, that have propelled our survival to a large extent, in such a way that by 2050, 2 billion more people will be there, right? Which is good for that people in a way. But as I was saying, it's also creating a lot of problems. And the success of medicine has been staggering, that's no doubt about that. And in many ways, when we think about disease, we tend to think in those great successes in a kind of a simple way, right? The development of vaccination was something that was a follow-up of our understanding of what was infection, what was a pathogen. I recommend to all of you to look at the history of that, because it's really fascinating and non-trivial, right? Once you know the answer, it's like, oh, of course, you're right. There's an infectious agent, but it's not so simple, right? And developing vaccination gives you this linear causal perspective, like I have a pathogen, I have a vaccine, right? And somehow this is kind of the solution to the problem. For cancer, which again, it has a very interesting historical record of changing perceptions and understandings. I remember many years ago when the molecular biology of cancer came about, and it was this beautiful idea that cancer is caused by some genetic, in the end, some genetic causal agent. There are genes that do not function in some way, and so maybe the target of that is going into the genes, right? This is something that 30 years ago was very much the state of the art, both in physics and in biology, the reductionist approach, right? I go into the basis, into the minimal potential elements, genes, for example, for biology, and for physicists was particles, right? And there were, I remember physicists saying, if I understand particle physics, I will understand everything, because in the end, you know, chemistry is a particular case of physics, and biology is just a particular case of chemistry, and so on and so forth, right? This is a complete mistake, of course. But what we have learned is that, right, there are genes, we need to identify them, and that's a big effort nowadays. But genes interact, and cells interact, and that makes a big difference, right? And also, as I will point out, cancer involves more things that we have to take into account. And in brains, what about neuroscience, right? Neuroscience has always been, since almost the beginning of the 50s in the last century, a system science, right? With the rise of cybernetics and computers, people started to make models of neural networks, right? And since then, the idea that you need to look at things interacting, that you're not going to go too far just looking at the single neuron, right? That doesn't mean that it's not important, but it's not enough for addressing learning, memory, consciousness, etc., and disease. A lot of things have happened. And also, at some point, there was kind of the magic bullet idea that you have some psychiatric disorder or some psychobiological disorder. I can maybe find out the right drug, and the drug solves the problem, right? And again, this kind of linear causality is not working. I put this book, Awakenings, because it's an extremely inspiring book. If you haven't read it, I recommend. Not because Oliver Sacks, the author, this is the movie actually, right? This is a book for that. Not because Oliver Sacks is a reductionist. He wasn't a reductionist. In the book, he explains the story of the patients that were completely frozen, right? And that he used L-DOPA, so a drug, to actually completely change the state of the patients, right? And that could, again, the partial success of the story will suggest that I have a problem, I have a target that I can identify in a very simple way, but that's not the case, unfortunately, most of the time. Well, this course that we teach involves a number of different case studies. We explore mainly immune diseases and virus-related diseases, right? So there's a number of things that I will mention you. We also explore cancer as a complex disease, right? And we also do a number of interesting things about brain diseases and how a number of tools from complex systems like networks, in particular language networks, can help to actually get insight into brain diseases. Since brain is such a simple problem, I'm not going to go into that today. I'm just kidding. And I want to raise some other points on something we are doing that I think that you will find interesting. So the first thing, because you use too much this word, complexity, complex system. What the hell is a complex system, right? And you will find that probably people saying, well, we really don't know. People who said, isn't it everything complex, right? So I want to make the effort of bringing what people working in complex systems, we believe it's the kind of the general principle, right? Complex systems display emergent properties, right? Meaning what? Meaning that you have a system made of many parts. That could be the cells in in a musculoskeletal kind of context, building organs and tissues and whatever you want. Can be neurons in a brain, can be, for example, one of my favorite examples can be social insects, right? Like ants or termites. Why I'm using this example? Well, look at this. You know, I don't know if you have seen a termite in your life, but a termite is an extremely simple and dumb insect. They are blind, they are very small on the millimeter scale, right? They communicate using chemical signals, and now, despite all this simplicity, the collective, the colony, creates this huge nest, which can be as high of the roof of this room. So this 3 orders of magnitude larger, with a huge architecture, with an agriculture system based on fungi, a ventilation system, etc., etc., etc. What is the knowledge for building that, right? As much as you can spend your entire life looking at single termites and knowing a lot of them, about them, you're not going to get any clue about how that happens. The nest, the society, the structure that the society builds is an emergent property. It's an emergent phenomenon that comes from interaction of many individuals, right? The same happens in the brain. So if you want to understand memory, consciousness, the way we treat information that comes from our senses, looking at single neurons is not going to work, right? Because it's not the correct scale. In summary, you have complexity because at some scale, when you analyze the properties you want to understand, there are features, there are properties that you cannot reduce to the properties of the elements at the smaller scale, right? It's not possible. So that doesn't mean that everything is going to be a mystery. It's just that on the new scale, you need to take into account interactions. And interactions is what makes the difference. All right. So there's a number of contexts where we make sense of this, making theory. In physics, there's been a lot of efforts into building theories for complexity, in particular in systems that display phase transitions. And I mentioned that, you know what a phase transition is. You take water, you take ice from the refrigerator, you heat it, at some point the ice becomes water, you have a phase transition, and if you keep heating this, at some point the water becomes steam, right? That doesn't seem extremely exciting, but it turns out that many systems that we can analyze, and that includes the way diseases happen, can be understood in terms of phase transitions. Well-defined phases where I can see properties that are very robust and are very different from phase to phase, right? And that's one of the things that we also develop in the course. So why complex diseases? Well, why not everything is simple? But you will not be here enjoying Barcelona if that was the case, right? And the reason for that has to do with a very fundamental idea that has been briefly mentioned, the genotype-phenotype map, right? If things were simple, we will go into the genes and identify the list of the genes that do functions, and we will know what will be the outcome of that, right? I have this list, that's my organism, okay? That would mean that from the genotype to the phenotype, right, the organism, it will be a linear connection. Unfortunately, that's not the case, right? Well, it's not unfortunate because if it were the case, we will not be here either. The plot I'm showing here is kind of an intuitive picture of the whole idea. It's a fitness landscape, right? It's what we use in evolutionary biology, but also in evolutionary algorithms and in the new wave within machine learning. It's this multidimensional space where you can actually find out solutions to our problems. And typically, for a complex problem, our fitness landscape, that in a way defines the potential optima for a given problem, we don't have one solution or a few, we have many possibilities, right? And that's what we call the nonlinearity in the genotype-phenotype mapping. And that's why, essentially, we are here. So, as I was saying, the idea, the philosophy behind the kind of things I'm going to discuss with you is something that has to do with 2 very important topics. One is scales, and just mention briefly the example of the ant colony or the termites or the brain, right? There are different scales of description for a given problem, okay? And what happened when I move from one scale to another? What's missing? How much detail I need to keep from the lower scale, right? In a way, this is the question: what is the right scale to understand a complex system? And it's not a trivial question. What is the use of a model? Why do we use models? I don't have to convince you that models are relevant, right? But I want to convince you that simple models can be also relevant and insightful, okay? So, can simple models explain disease states? So, let's start with, I'm going to go for 3 case studies, right? And I'm going to compress a lot of information. So, I'm going to try to be intuitive in the ideas. First is viruses and virus-related diseases. The second will be cancer. And the last one will be something we are working on, has to do with disease, health, and on a planetary scale, and how this can be tackled with the same kind of approximations. Well, in this diagram, I try to put together the basic idea within the context of virology of these different scales. And in these little boxes here, I use examples of mathematical models that correspond to the given problems that every single scale addresses. What kind of problems can we look at when we look at viruses? We can go from the simplest scale. The simplest doesn't mean that models are simple necessarily, right? And a lot of work on how viruses get assembled, the molecular level, involves molecular dynamics and sophisticated computer approximations, right? That on the smaller scale, we need to understand how viruses get constructed, right? If we move up, when you have populations of viruses, the relevance of that, the relevance of how they assemble just vanishes. It's not that important, right? We move into a new scale. So the questions now are different and have to do with the presence of mutation and selection. What happens when these viruses interact with us, with our body, right? They interact with a given context, and these 2 years of COVID have shown us a lot of interesting new challenges. And each tissue it's kind of a new context where the virus can thrive or not. But these tissues and organs belong to a whole organism. And so we have to actually have into account this idea that maybe having a more systems approach is relevant, right? And if we keep moving up, then we are not looking into the organs of an individual, we look into individuals which interact within populations, right? And so we have the classic work in epidemiology that has been, unfortunately, so popular in the last 2 years for everyone. And we can actually scale up into the large scale, into the global problems, right? Pandemics, and that allows us to use other models. Let me illustrate a couple of examples of why these simple models can help to actually solve things that are kind of even mysterious, right? This is one of the examples we study in this course, is the development of AIDS within patients, and the puzzle that represents, this is an old case study, that represents what's called this asymptomatic phase. In a nutshell, the idea is people getting infected with HIV, looking in blood, look at the amount of the viremia, so how many RNA copies of the virus you detect on blood. And what you will see is that it's a peak initially. The scale, the original scale is weeks, right? It's kind of a standard infection as an immune response to the system. And then essentially it goes down, right? That's kind of a typical diagram. Then look at the x-axis, the scale changes for years, right? And over these years, what we observe is that that's before the treatments, right? What we observe essentially is that the virus is kept at bay. So it's a very low concentration on blood, right? At some point, this changes. It starts to raise rapidly, right? It could be even exponential. And with a decline and collapse of the immune system, you have a huge amount of viruses there that actually lead into the what's called to the AIDS clinical situation, right? So why this latency? What's the origin of this phase where it seems that nothing interesting happens? Just to point out that because we have actually seen this in all these 2 years, all the crap that can come from the media and from highlighting things that are scientifically nonsense, is that some people, and that included some scientists, said that, look, the virus seems to be under control. Maybe it's not the virus which causes that, right? Maybe it's, and then it's all kinds of stories, right? That was, of course, were not correct. And you actually look at the historical development of AIDS, you'll see also that this all the conspiracy thing of that was created in a lab and then blah, blah, blah. Right. So what happens here? And the solution to that came from 2 sources. One from a very simple clinical test, which is let's start trying, that's again the linear approach, some drug that blocks the expression or the production of the virus, right? That's something that we know now. It's transient. The virus eventually evolves into something that escapes from single drugs, right? But when people try it, what happens is that you see effectively that the virus production is blocked. And in patients, you'll see a decay in the number of viruses, right? That follows what exactly you expect from a very simple mathematical model, which essentially says that if you are unable to reproduce, you're just going to die, right? And what you expect from that, you probably have seen that in some of your courses, is exponential decay. So this can be fit very well, right? And so what kind of things, what kind of message this gives us? Well, the good thing is that you can make a very simple model, extremely simple model, and in the end you just have one equation that gives a prediction of how the virus will decline if he just cannot replicate anymore, okay? There were 2 papers in Nature that had an extremely important impact in the area. This one, where some of my colleagues of the Santa Fe Institute, like Perelson and Abid and Neumann, were involved, and another one that was published by the group in Oxford, where essentially they do 2 things. One is actually show this kind of experimental outcome, and the other thing is use this simple mathematical model, right? And the simple mathematical model was able to actually provide a measure of one parameter that was extremely important. How rapidly is the virus removed from blood, right? Because this decay in the virus, in the end, is reflecting how efficient is the system for cleaning up this variant that I have there. And the message was very interesting. If you make the little calculation, which we do in the course, is that in about 2 days, the virus has been cleaning up. But what's the meaning of that? The meaning is that this is the time scale of something, right, which is not years. It's a time scale that says that the immune system, and my guess is that you know something about immune systems, you have a pathogen, you identify the pathogen very specifically, and you have an explosion of cells that explicitly go and target this particular virus, which means it's a cost for your immune system. It means that on a scale of only 2 days, you have a huge response in cleaning. But that means that if the virus is able to escape from that, and it does, over these years, what you'll see is that there's a huge effort by the immune system of fighting the real virus. So with this apparent calm that seems that nothing is happening, is a huge arms race. And the arms race is damaging the immune system more and more and more. And that brought into the next level, which also received modeling efforts, where essentially is that knowing that the virus is interacting with the immune system and clean up in a short time scale, what happens if I make a model where I introduce kind of simple evolutionary rules where new variants emerge? And the prediction of the model, which essentially seems correct, was that as soon as the number of different viruses starts to accumulate, the different antigenic diversity, right, the immune system is not going to be able to react to that and will collapse, okay? So the first glimpse that we have that this was the case was using this very simple mathematical model. So another question, You have heard, again, all over these 2 years that RNA viruses, COVID in particular, mutate very fast, right? Sometimes this is misused in the news, like, oh, this virus mutates so much. Well, yeah, we mean, this is normal, right? That doesn't mean anything necessarily. But this is a legitimate question. For a virus, for an RNA virus, it's good to mutate, okay? It's not good for us. Our cells actually have a huge machinery that is fully dedicated to being sure that your cells are not going to replicate in any wrong way, right? And that's why this great evolutionary invention, programmed cell death, right? I don't know who said that, something like, every day all your cells wake up thinking in suicide, but somebody makes them calm down and not do it, right? What does it mean? It means that every single mistake, right, can send a signal to the molecular machinery to say, okay, you die because you're a danger for the system. But what about RNA viruses? Viruses want to do the opposite, right? If I'm not mutating fast, the immune system can identify different variants of the virus and just remove it. So it's a good strategy to mutate because you can actually co-evolve with the immune system. So these two guys in the 70s, I just wanted to illustrate why models can be so powerful. In the 70s, Manfred Eigen and Peter Schuster, I know Peter, he is one of the most smart persons I ever met in my life. They were exploring a problem that is more theoretical. It's a problem of related with the origins of life and the early evolution of life. If you have a population of molecules that replicate, and when they replicate, they make mistakes because mutation is inevitable unless you have a machinery to repair. Is there anything that we can say about the impact of mutation in population dynamics? And they found out something really interesting, right? They found out that using a mathematical model with molecules that just replicate and make mistakes, there's a critical mutation rate. There's a mutation rate beyond which information cannot be stable, right? Anything that you have in the system that you can say, this is advantageous, this is something that is good for you for replicating, is lost as soon as mutation goes beyond that threshold. You might have gone into that without knowing, if you have been doing evolutionary modeling, genetic algorithms and things like that. That's sometimes that happens to my students, that they do a model that evolves something and say, I don't know what's happening, it's not working well. And one of the reasons is that the mutation rate you're using, right, it's too high. Once the mutation rate goes beyond some threshold, things are not going to be stable in terms of Darwinian evolution. The prediction from the model of Eigen and Schuster is that there's a mutation rate, a critical mutation rate, that goes as the inverse of genome length, right? So in my genome is, say, HIV, 10,000 nucleotides. The maximum mutation rate that can be achieved without losing information is 1 over 10,000, right? A very well-defined prediction. At the time that was done, in the 70s, there was no idea about what was the mutation rate of viruses, right? So it was kind of a bold thing to say. But the thing is that as soon as we were able to actually manage that, to understand what was the mutation rate of RNA viruses, the prediction from the theory fits very well, right? There's a critical mutation rate, meaning that if you mutate even a little more, you are just extinct, right? You cannot deal with that anymore. And that's important. Thresholds are very important, right? This is nonlinear thinking. So the idea here was that you cannot think in viruses as kind of a species, but quasi-species. You have a mutant cloud of viruses, right? Which we know is the relevant part of the story, and there are critical values for that. What's the use of that for thinking in disease? And something came about to several people in suggesting the following. And that's something that is right now very active because we are identifying finally the right drugs for doing that. And I'll explain you in a moment in a different context why this is also important. What if we were able to increase mutagenesis a little more, right? I have, for example, in vitro, I have a population of cells I have HIV viruses infecting them. What about if using what's called nucleoside analogs? Doesn't matter. Something that allows us to push a little more mutation rate. Consistently with the prediction, right, you induce an extra lethality that just wipes out the virus, right? That, of course, is not trivial. You could say, okay, let's go into the clinical essays, right? And that's, of course, the dream in the end. But as you know, or you will in some years, right? Everything in biomedical research is a long story. When I was a student a long time ago, I remember that in those lectures on molecular genetics, it was very exciting. Everything was kind of, wow, immunotherapy for cancer, gene therapy, right? Everything seemed close, right? We needed 30 years to make that really happening, right? It's happening now, okay? But took a while. Moving scales, right? Let's go up. In epidemics, epidemic spreading. One of the important results of mathematical models of epidemiology is a classic result, right? Which pervades the fact that vaccines can work and pervades the fact that nonlinearities are so important to understand the real world, is epidemic spreading. This is a diagram that I used in a course I was teaching, in a lecture, I was teaching within a course of virus dynamics years ago. I taught that year after year. And I put it here because it's a reminder of things, right? This is March 2020. And you can see this is kind of a diagram with a timeline of big pandemic events, right, that have wiped out a lot of people. And COVID-19 is there. Like, it's ongoing, it's small, we'll see, right? Unfortunately, we all know what happened with that, right? It's a good reminder of the thing. And also, I just want to point out that here there's a number of important things that interact together, right? COVID-19 is a reminder that we are invading our planet and making more and more easy to actually make contact with viruses and other pathogens that usually will not be in contact with us, right? But are now more and more likely to happen. That and globalization of transport makes a difference, right? And since I can remember, new viruses have been emerging. Many cases that we've been able to control them, right? But we need to be aware that this is not going to happen always. And in the end, let me say that COVID-19 has shown that we are fragile, that we cannot give for granted that everything is going to work well as we expand in our planet, right? And that in the future, that might happen again, and that you have been lucky. I mean, COVID could have been much worse. So, many new emerging viruses are likely to happen. This is a map of things that have been happening all over the place for the years. Many of them you don't even remember. Like before COVID-19, there was some outbreaks of SARS-related viruses that were kept under control, right? And that time, this time we have been so lucky with that. Two important messages that have to do with mathematics of epidemics. The first is, if you don't control the thing on time at the beginning, you are not going to control it. Meaning what? Meaning that the first outcome that we know from the models, the first nonlinear outcome we know is exponential growth. Right? Something that you need to explain to politicians again and again, right? The world is not linear. And exponential world means that, growth means that if you are a self-replicating entity of some kind, at the beginning, you might be under small population numbers, right? But growth is exponential, and eventually you escape control and you become a pandemic. And the second thing is that this nonlinear character of epidemic spreading, right? The diagram on the lower part is a very simple diagram where essentially for measles, right? What's taken here is taking populations of different sizes. You can use urban populations usually, but it can be also on islands. and see how likely is that you observe measles in the population, right, over the year. Here is just how many months you see that there. And what the diagram shows is that if the population is low enough, right, you are not going to see the virus. The virus has a really hard time to really spread and go anywhere. But if you go beyond a threshold of population, right, you can actually see the virus there, like somewhere, something that is already there in the system, and you are not going to remove it. And it's a very nonlinear behavior. That means something that goes into the intuition that more population, more virus, but it's not the case, right? It's a very important thresholds that makes this work. And the simplest model that we use, but it's not the only one, right, is the SIS model, which assumes that you have population of 2 types of people, right? Susceptibles, so they're not infected and infected, right? In this very simple scheme, what essentially we consider is that there are 2 processes. One is that if I interact with someone that is infected and I am not, I can get infected too, right? And the process of infection, again, in this parameter mu, it's kind of a reaction, right? I collapse a lot of information. A lot of molecular biology is there as a single number. Remember the idea. I move to another scale, and I can ignore a lot of the molecular and cellular and tissue features, because at that scale, they are not important, right? And the second part is that I can get healthy again after some time. Right? This is the simplest model I can formulate, but the good thing of this very simple model is that it actually provides a rationale for this threshold phenomenon. Below some infection rate, you are not going to survive. You need to go at least beyond a critical number. Again, critical numbers, right? And phase transitions. And that also makes sense for understanding why vaccination works. This is a non-linear phenomenon, and you can actually show mathematically that depending on the infectious efficiency of the virus, if I push a little bit the population in a way that a fraction of the individuals are immune to the virus, right? So I just move them out from the potential infection, I can show that the threshold that I just showed, it's moving. And essentially, again, in a nutshell, is that if I make the things complicated enough for the virus to propagate, I just can wipe out the complete pandemic, okay? There's a number of ideas that come out from this, because that was a very, very simple model. And for example, what about this threshold when I'm considering a disease that is much more complex, right? I just use 2 compartments, but depending on the kind of the pathogen, the number of compartments I have to consider is different. Sometimes some compartments can be peculiar. For example, corpses. When I consider Ebola, in Ebola, corpses keep the virus being infectious, and that was a big problem because many people use as a social ceremony to touch the diseased, right? So they got the virus. But the good message is that for any of these case studies, there's always a critical threshold for propagation, right? And you can identify it, and you can actually measure. With the early revolution at the beginning of this century, with networks, a lot of things happen within, not also in epidemics, but in molecular biology, in cancer, in networks, in all kinds of things. Our understanding of epidemic spreading has also improved a lot, taking into account the mathematics of networks, right? How actually they propagate through populations, maybe through cities, through continents, etc., etc. A lot has been understood. And one of the big things that happen, 2 important things that we have understood, is that networks, the fabric of propagation where the viruses move, have 2 different things, 2 important things that we didn't know before, right? One is that they are small worlds, right? You have probably heard about that. Just briefly saying, we all have this idea of that the world is small, right? You go into in a train, you take the train, and there's someone sitting just next to you, right? and you start a conversation. I don't, because I don't like people, but you start a conversation, and all of a sudden it's, oh, look, it's the best friend of my brother at the university. Oh, isn't the world small? Well, this is not anecdotal. It's true. The world is small, right? And that has a lot of implications, which means that, in particular, that information propagates very efficiently, very easily, right? And you have heard about these 6 degrees of separation. If you made the network of all the people you know, right? Let's say friends, okay? You have, I have some friends, and I consider myself connected to them. Usually they are connected between them, right? So they are triangles. And they are connected to other people, et cetera, right? Okay? Those of you who have no friends might not understand what I'm saying, but the idea is there's a network of social connections, and if you make the experiment of moving from one node to another node, just chosen at random, there's 6 degrees of separation in a country made of millions. That was analyzed by mathematicians and physicists, and this is a kind of a simple way of presenting this. Imagine you have your city is a network, a square network. Every single node could be you, for example, and neighbors. And you do the experiment of saying, right, but things are not just geographic, right? I know someone else from somewhere else, from a distant part. So what happens if I connect things at random, just a few of them, right? And in this rendering, what we do is to make these random connections and make them smaller as the original ones, right? So I get something kind of a scrambled lattice. And the thing is that with just a few of these random connections, this lattice, where at the beginning I needed to walk step by step to go somewhere, is a small world. It's very navigable, very easy to navigate. And the second thing that we learn is that these networks are also scale-free, that they are extremely heterogeneous. This is a map of the internet from 20 years ago. It's been expanding hyper-exponentially, right? So here, every node will be this kind of network, this kind of plot will be either a computer, right, or a server. And what we know, nobody well now, is that most of things in these webs have a small number of connections, but some of them, a few of them, are huge amount of connections, right? So these are big servers that make the network very navigable. We move in the network very easily because of them, right? But also fragile. So if they go down, everything goes down. One of the interesting outcomes of this is that when we map the protein interaction networks, what we found out is those proteins that have lots of connections are, surprisingly or not, those responsible or typically mutated in proliferative diseases like cancer, right? So the hubs are also there, and they have an important role. Just to point out, for those of you who don't know, that kind of ideas has been used to actually define what's called the human disease network, which we don't have time to talk about, but anyway. So, just to finish this part, what happened is that when we look at the real world, right, what we find out is that the network where some of the diseases propagate is scale-free. It's also very heterogeneous, right? And that makes a difference because the transition that I was describing before disappears. But more importantly, because, for example, for sexual transmitted diseases, you can actually do these diagrams, making sure that you actually map these networks and see that in these cases, propagation is highly dependent on these nodes that have a lot of contacts, right? And that you can actually defeat the virus by controlling these few nodes, right? Which actually makes a big difference in terms of thinking on the problem, right? Okay, there's a lot that has happened over the last 20 years, and in particular in the last 2 years, in terms of making powerful, predictable models of epidemic spreading on all the scales and having into account all the relevant information from omics to the transportation networks, right? And this is a cover of Time magazine I used to use in this lecture I gave on epidemic spreading, which was unfortunately pretty much accurate, right? That we are not ready for the next pandemic. Okay, an important message I want to stress, that I think this is very important and should be clear for any people starting a PhD, but sometimes it's not. And is that a given model is right for what, right? This is a very unfortunate sentence that I hate very much. It says something like, all models are wrong, but some are useful, right? Okay, I think that is sheer stupidity, because you can say that about almost everything, right? The models are right if you have the right question, right? And I just tell you that my students, when they start their PhD, the idea is that they have a big question in mind, something that will be the achievement of a PhD if everything went well. And then the steps that brings you there have interesting questions that you need to solve. But you need to have a question. What are you looking for, right? So, in my humble opinion, I mean, if you are asked and you say, well, I want to make a model of, no. What is your question? What do you want to solve? The model is the way you're going to solve it, right? It's a different thing. So what about cancer? Cancer is a big trouble. You know, it's been, I remember that many years ago there was this war against cancer. Cancer is going to be defeated. So I don't like all this war-like narrative. But why is cancer still there, right? Isn't it in the end something that grows inside you? It should be simple to solve that. Well, the problem is that cancer is a complex system in itself. And the right way of pointing will be kind of saying in plural. I mean, it's not one cancer, it's many different diseases, right? But it results from a number of things that we also explore in this course, because it's aging. We live too much. That's one thing, let's face it, right? Evolution has prepared us for having kids in the 20s and then die because, you know, some lion kills us or something like that. But we survive. We survive because, again, we have medicine and everything works well. Good for me, because, I mean, I use glasses, as you can see. So, yeah, if I had to hunt anything, I probably will kill someone of my tribe, right? So I don't need that, no problem. But we live a lot, and cancer is the outcome of a conflict between two tendencies. We are not machines. Our bodies are not machines. We have this major difference, which is that is proliferation, regeneration, is a constant maintenance of the system to keep it alive, right? Which has a lot of advantages, but the tension between growing and controlling growth, right? That's something that actually pushes our own body into this kind of critical state, right? And any imbalance goes into the wrong direction, right? If genes that have to do with making things proliferate, why is that? Because, for example, if we lose cells every day, right, we need to replace them, right? So we need that. And they're the genes that stop things replicating. So which are the genes that control things like the cell has double the chromosomes and everything. It's going to replicate, but it detects something that is wrong, right? So stop. Don't move. Let's, first of all, check out everything, right? What happens if anything of this fails? That the key rule of our bodies is our cells cooperate. We are a system, right? Darwinian selection is forbidden. There cannot be Darwinian selection. Not the rule of survival of who replicates faster is not part of the equation. As soon as this is broken, you can have problems, right? The fact that control mechanisms are there and that work well, it can be seen by many ways, right? In the course, we look at that in different ways. Let me just give you 2 examples. One is the understanding that we know now that many cells in our bodies have key genes mutated. They should be cancerous cells, or they are not, because we do have cancer, right? But this is at bay. And the second is that you can make a small calculation and get a lot of insight from the following problem. Imagine that you think in the following. You know, we always say we share our genome, most of it, with our close friends, vertebrates, primates, but also with whales and dolphins and everything. Whales are very large, you know. It's 1,000 more cells than us, okay? So for elephants. Interestingly, you can make a simple calculation saying if everything is preserved, as we know, it's conserved in terms of the machinery of mutation and replication, etc. What would be the probability that a whale has cancer? 1. So essentially, all whales should be dead of cancer floating in the ocean. But of course, it's not happening, right? So what tells us this? This tells us that there's something in these animals, and you're learning a lot about that nowadays, makes protection against cancer 1,000 times more efficient than us. How? Right? You see, looking at some particular case studies, you can gain new questions to formulate. Okay? So, importantly, cancer is an evolutionary disease, right? Cancer is so successful because it's not just growing. It's not just an accident. You keep growing. Cancer takes advantage of the fact that in a genome that is free from control, you can actually do what evolution does on a very short time scale, which is finding out ways, as the evolutionary algorithms that you're maybe using for some applications, to solve problems. And for example, I'm a tumor, I've been growing, I cannot grow anymore because there's tissue around me that is making pressure, right? What can I do? You need innovations, right? No problem. If there's a mutation of one of the genes that is involved in the formation of the placenta, you see, totally different story, right? You can actually get blood vessels, right? And you keep going. So you can actually go through a process where evolution is important. A lot of ideas from ecology and evolution are being used nowadays in cancer to rethink the way we consider disease, right? Why is that? And that's again, some things that we consider in the course. Why is this important? Well, we again see phenomena that we need to understand, right? One of them, for example, I tell my students, something that is in the literature, and sometimes even the physicians look at that like in a strange mood, right? But now it's been taken very seriously, which is spontaneous regression, right? You have these cancer cases, you have metastasis, you should not live more than 3 months. And so you go to your house, you start to think, okay, let's put order in my life. But after 3 months, you feel well, and 4 months, you feel well. What the hell? So you go to the physician, you're checked, and there's nothing there, right? So what happened there? We know that the immune system has been able to react in a very powerful way, wiping out the whole cancer. How is this happening exactly? We don't know yet, right? Immunotherapy, which is probably you have heard, is probably the best weapon we have now against cancer, is showing that this is the immune system, the key, okay? And a mathematical model, not showing you that here, shows that there's a mathematical reason why this works. And the other problem connected with Darwinian evolution has to do with the way we treat cancer. Cancers are very heterogeneous populations, right? Because of mutation, you don't have a single kind of clone. You have a lot of populations there. That means that there's a lot of opportunities of cancer for responding to aggressive therapy. And in some kind of cancer, for example, glioblastoma, what happened is that the typical scenario is that you can reduce the tumor, right, with chemotherapy, with radiotherapy. Now things are changing again with immunotherapy. But the typical scenario is that, yes, but someone is surviving there, right? And the survivors are the most aggressive and resistant. The tumor goes back and kills you. So we have learned a lot of things about that in ecology. When you have a pest in a population in the field that is aggressive against your crops, being extremely aggressive against the pathogen, right, the insect or whatever it is not working well because it works just briefly, but the resistant clones, the resistant variants are going to give you a very hard time, right? It's much better to be not so aggressive, trying to have having the trouble there, right? So that it has a hard time, but not selection, not put selection in place. Selection means that you select for those who are more aggressive. So new strategies are kind of rethinking that in terms of, for example, what about making the cancer a chronic disease? So maintain it there. Also in cancer, we have these multiple scales. I'm not going to go into all the details, but for every single problem that you can formulate, for every single question, there's the right model and the right level of description, right? From just the pure mutations and having an omics perspective of what is mutated in tissue samples, to what happened with chromosomes. That is one of the most fascinating things of cancer. When you look at one of your cells, a healthy cell, you have this 23 pairs of chromosomes, everything more or less equal, fine. When you look at the typical cancer cell, everything is scrambled. It's a huge chaos. Chromosomes are cut in pieces or duplicated, or how is it possible? I mean, why the cell keeps working, right? Something that we suspect has to do with something I'm going to tell you in a moment. And if I go up, right, I go up and I go into different levels, different kind of things are required from going to one level to another, and new questions emerge, right? Okay, so one of the things that we did some years ago, and I wanted to just briefly mention because it connects with the virus problem, and I want to point out that actually one of the interesting things of having a whole perspective of complexity is that you can actually get insights from one discipline to another, right? And use cancers from one place to another. One of the things that we were interested in is precisely this problem. So if cancers can be unstable, right, and scrambled genomes and et cetera, how much unstable can they be, right? It cannot be infinite. So you can actually see this instability, this disorder growing looking at the pathological samples, right? For example, for colon cancer, if you look at different degrees of cancer development, you will see that if you make histological samples, you'll see that you go from the ordered structure at the beginning, we have these colon crypts, into something that is more and more and more disordered. And what we did some years ago with his colleague Tom Bisboek from Harvard is to actually make the following idea, conceptual idea. RNA viruses are not cells, okay? But they also have this property of being unstable in the sense of having a high mutation rate. RNA viruses live on the edge of catastrophe, right? They have the highest mutation rate compatible with keep going but not losing information. Could it be the case for cancer cells, that they are very heterogeneous populations with a lot of instability in their genome? So we proposed this idea. What is this error catastrophe, this threshold of information that we see in viruses applies to cancer? So we developed several models. We've been working on that over the years, suggesting that the idea that maybe disorder brings the cell into the simplest thing compatible with a given context, right? And in that context, not going into the details, but we can actually predict that there's an instability rate, if you want a maximum mutation rate, compatible with rapid replication. But as soon as you start to increase this instability, you rapidly go down, right? So the message here will be that cancers also might self-organize themselves into this critical boundary. So they work very well, they are very efficient in terms of evolution, but they are fragile if we increase instability. And that's the idea that we actually brought. We not only use mathematical models, but computational models where every single cell carries a digital genome, a very simple genome with genes that do different things, right? Raw, mutation, stability, et cetera, et cetera. And we predicted that it has to be a threshold. Even with different models, mathematical, computational, there has to be a threshold. If you plot the whole landscape, here what we have is a projection of this multidimensional space. Have in mind that even for these digital genomes, right? If I have, 64 genes, which is not a big number really, but you have, if every single gene can be mutated or not, is 2 to the 64. So the space of search is huge, even for that small system. So what we did was characterize every single genome with a fitness function, right? How rapid you replicate, and put these connections, right? Which actually says how likely is that by a single mutation, I go from you to you, right? In my simulation. So the landscape is extremely complex, right? But nevertheless, have in mind that if you think in mutation as some global attribute, it's a coarse-grained attribute, right? Despite all these complexities, the message from the model is if you increase mutation rate, if you make cancer cells a bit more unstable, you might remove them from the equation, right? Is that the case? Well, over the last 5 years, a lot has happened, and there was already a lot of indirect evidence for that. Some kinds of cancer that we knew that were more unstable and were treated with chemotherapy, they were easily removed from the system, right? Suggesting that the more unstable you are, the more trouble you can have. And in the last 2 years, there's been a number of new papers suggesting that this might be the case. And we needed to wait to have the right drugs that are not cytotoxic to actually get there. So we'll see. I hope our prediction was correct. And my last part, you'll see a totally crazy story, right? But we are here. You cannot stop me. So what about disease in a planetary context? It sounds strange, I know, right? But this is not speculation. One of the reasons I thought it was nice to bring this here is that Pompeu Fabra has this very ambitious initiative of building a center for planetary well-being. Meaning what? Meaning that the more we know about how things happen globally, the more clear it is that having human disease in the right context needs the planet as part of the context. And we need to address problems in a much more general perspective. So the center will be built. Hope a lot of interesting opportunities are going to happen. And complex systems will be a very important part of the study. So what in particular I'm interested in, and that we have been addressed over the last 6 years, right, in my lab. Well, it's a whole story behind our understanding of ecosystems that has been growing in the last 2 decades. And in particular, trying to assess what's going to be, and prediction is a very important problem here, what's going to be the impact of climate change in our lives, right? Very unfortunately, right, I don't like to give a grim message at this time of the morning, but unfortunately, the worst case scenarios predicted by climate models are those that are happening, right? So when we talk about climate emergency, it's not a metaphor. It's something that we need to address right now. And one of the issues that comes from studies of field data and models is that very unfortunately, in the future, changes, and that's a typical property of complex systems, are not going to happen in a smooth way. So we might dream that, biodiversity declines, temperatures get higher, but maybe we can adapt to that. That's the message that some politicians use, adaptation. They don't know how, but what the hell? We just use the words, isn't it? We will adapt. The problem is that changes are going to happen in non-smooth ways, and probably in rapid shifts, okay? And that means several things. One is that, on the one hand, for example, temperatures are rising, going up, right, as CO2 concentrations keep going. Is this going to be smooth? Sometimes you look at the newspapers and say by 2000 and at the end of the century, we'll have 6 more degrees or 4 more degrees. And it seems not good, but it's like, well, okay, I don't know, I can just have air conditioning. But the problem is that as soon as we also move beyond some thresholds, things will accelerate, and will accelerate out of control, right? More than 2 degrees is a critical number, not because it's going to be difficult to adapt, it's that things can be really bad, right? Because the climate has its own self-organizing properties, and temperatures can just go up quickly, and we are not going to adapt. It's no way of maintaining the civilization. So we need to do something. And extreme temperatures is the very clear situation. When I used this slide 3 years ago, there was this particular 2 cases that unfortunately are becoming more and more common. In practically the same month in the north of the United States was minus 50 degrees drops of temperature, while in places on Southwest Asia or in Australia in particular, in this place, there were 50 degrees temperatures that kill a lot of animals. We are not able to adapt to that. So what we can do, of course, I'm going to tell you about a science approach to the problem, but just want to make the point that I'm not saying that technology will save us. So, let's just don't worry much about that, right? But I do think technology is important. So there's this old idea of geoengineering. What about blocking the light of the sun, making more clouds, for example, to kind of repel, to kind of cool down the temperature. That doesn't solve the problem, really, right? Because CO2 will be there. But there's been there. Nobody knows if it was going to work or not, and several of these technologies have staggering costs. So what we proposed, right, it's something that some of you will immediately say, this guy is nuts, but the following thing. What about to engineer ecosystems, right? Using what? Using something that my lab is developing with this synthetic biology. What about considering the following idea? If I want to engineer something like in geoengineering, for example, one of the strategies is to make millions of small satellites with mirrors, right, that you can put in space and deflect the sun of the light of the sun. Might sound crazy, but it's there. It's one possibility. But you need to create all the machines. We can make these huge factories of creating steam to make clouds, right? But you need to create these machines, thousands of them. What about taking advantage of biology? Biology involves something that is really interesting, right? Biological entities self-replicate themselves, right? So what about, and that's the idea of bioengineering the biosphere, what about modifying something that is there in ecosystems, right? In such a way that we can exploit the fact that if it brings part of the solution, it replicates itself. So the little machines are alive and scales up with the problem, right? That's the idea. So the literature is not big because we invented the idea, so it's just going. But more and more people is paying attention to that. And interestingly, people even for conservation biology, which is the people that immediately says, this is not something we should do, right? So the idea for this, we call terraformation. Terraformation means that you probably have heard this concept from, I don't know, Elon Musk. This idea that we will go all to Mars. How is something that escapes completely to my imagination. And especially have in mind that Mars, it's going to be great when a mission goes there and explores the planet, right? But Mars is there to killing us. I mean, let's face it. It's not an alternative. Terraformation is the idea that I can transform a planet to make it more, habitable for us, right? So what about terraforming our planet? The idea is create a synthetic organism that is able to spread. So I'm not hiding anything, right? It has to be delivered and spread. And this has to be tested, and I'll tell you a little bit about that, because now we seem to be in this state, and see if this is scalable, so if this is going to work on the large scale, right? Because it's not so easy. You have concerns about that, but have in mind the following. In synthetic biology, what we do essentially is to manipulate by advanced genetic engineering cells, adding new functionalities, new genes, whatever is needed, right? And that means that, for example, we can do something that is still a work in progress, but it's been one of the interesting proposals. For example, imagine you have concrete cracks, right? That's a very important problem for concrete infrastructures, right? Which is a very, very important thing. Exposed to the environment, you eventually get cracks, and the cracks go into the internal structure of the infrastructure and essentially collapse. What about repairing that? Repair is not trivial, but somebody suggested the following idea, which I want to use as a kind of a metaphor of what I'm suggesting. Imagine you create a cell, which is a modified microorganism that can live within the crack. It likes the crack, right? The inside, it's an acid environment, it's a basic environment. Many bacteria don't like that. But imagine that I modified it so that it can grow there, right? And create a mixture between a biological polymer and calcium carbonate, which is very hard, right? So as they keep growing, they fill the crack, right? And eventually the whole crack is filled. If I design the bacterium correctly, as soon as I'm getting out from that environment, which I'm okay for work, I'm going to die, right? I'm not going to be able to keep going. So that's why this kind of motif is called work and die, right? Sounds like some North Korean blema, but work and die. So in other words, if I design things properly, I can actually do a functionality, right, and be safe, right? And that's an idea that is going to be published in iScience very soon. That is actually a general concept. We know that things are going to change in the next decades, and 2050 is kind of a target for us of thinking in strategies. And one particular system that we have been analyzing, it's drylands. I know that when people think in biodiversity and all these things, you think in the Amazon rainforest, the coral reefs, etc., which are really cool. But almost 40% of habitats in the world are drylands. Don't think in deserts, right? Think in these places that have not much vegetation, not much rain. But the thing is that a third of the world population lives there, right? In environments that are under a lot of stress because of climate change, right? And in whole chunks of the world, if things keep going, they might jump from drylands, so you have vegetation, you keep water there, to deserts, right? And we know that happened in the past, they can happen in a short time scale. So what do we do about that, right? We published this paper in a paper in Science 1 year ago, and we actually found out that taking information from 60,000 places all over the planet, you can actually identify different thresholds that are going to be crossed by these ecosystems in time. So our suggestion, and we use that for different problems, from urban environments to drylands, right? The idea is the following. Imagine you take something that is already in the environment, in the soil. The soil, I know that doesn't sound exciting, but the soil in drylands is a quite fascinating ecosystem. You take, for example, a cyanobacteria, which are really important kind of organisms. You engineer it in such a way that it creates a molecule, right? It secretes a molecule that could be totally unbiological in many ways, that is able to keep a bit more moisture. And I stress a bit more because you cannot do miracles, right? There's not much rain there. But a bit more in a nonlinear system can mean a lot, right? So maybe you should confront a collapse in 20 years in the future. But maybe this engineering could create the conditions for pushing out this tipping point away, right? And we need time. So that's something that we believe could work, okay? So I know what you're thinking. Yeah, right, but what about the famous unintended consequences? And I think this is an important part of the story because, you know, even for a course where we think in mathematical models, etc., ethical issues are always there. And what about it? Yeah, what about it? Well, the thing is that since the 80s, when people get very hysterical about recombinant DNA and genetic engineering, and for some reason, and you can actually check the literature, some reason that people thought, are we going nuts? What is going to happen if we deliver something that has a modified gene in nature? Well, the first thing is that we cannot answer this question scientifically because everything was banned, right? No more experiments of that, so we don't have any experience. And the other thing is that we know in nature, bacteria are transferring information all the time, right? At a massive scale. And there's no Frankenstein coming out from the fields, right? That is a normal part of what's happening all the time. We do believe that is no such a thing as unintended consequences, especially if we have in mind that we are already doing everything wrong, right? So I don't think that should stop us from that, right? A little story I want to mention. I'm a member of the Santa Fe Institute in the USA, which is kind of the mecca of complex systems. And years ago, when Jurassic Park was being, kind of the plot was being prepared, etc., at the Institute, there were two people, two good friends of mine, which was part of the fathers of chaos theory. And Jeff Goldblum called to the Institute to ask, Doing Farmer in particular, how a mathematician working on chaos behaves. if you have seen the movie, I mean, no advice was taken, really. So, I'm finishing already. The idea is to test that. We've been working a lot on modeling, mathematical and computational models. Been working very closely with people that is working in these particular systems in the field, and we have a lot of support. And one issue that in particular I want to stress is that you can think that intervening in ecosystems has one problem for sure, is that ecosystems in a way are unique. So what happened with multiple tests, right? And the good news is that for the microbiome, in particular the gut microbiome, which you know it's a very important story in understanding complexity in health and disease, is an ecosystem, a very complex ecosystem where actually there's been a lot of work now to intervene, to use modified microorganisms to see if they can change the property of the system. So that's one of our multiple experiment system that we want to actually look at. A lot is happening. It's been recently done by a group in Israel, something that was quite unexpected and extraordinary, which is engineering E. coli, which you know is not an autotroph. It doesn't use light or anything from the environment like a plant. Has been engineered, well, has been forced actually to evolve into something that can take CO2 from the environment. It's still not efficient, but shows the potential for actually manipulating biology. And some good news is that we have been recently awarded by EMBL, the European Molecular Biology Lab, the most important institution in the world for molecular biology, that has made this step into moving into the large scale ecosystems and planetary health dynamics. And one part of the initiative is to actually support these experimental ideas of modeling and modifying ecosystems. So we'll start this year to have our first prototypes in this collaboration, and for those who might be interested, to open positions for a postdoc also. And we hope this is going to be the beginning of something. And again, multiple scales are involved. So when we are in ecosystems, as it happened with viruses and cancer, I didn't show you the diagram for brains, which is essentially neuroscience, the same story. Different scales, different ways of attacking the problem, right? But we believe, as I was saying, we need right now to do something that has, if you want, kind of the equivalent of what we did with agriculture, right? We modified nature for our benefit, of course, and we need to do it again. But, very important, preserving biodiversity, because without that, things are not going to go well. And I think that the major issues here is this question: what do we want, right? Can we not to do anything because we have this idea that things can be kept, preserved, even conserved, without modifying it? We have to be part of the equation. So we need to actually solve the problem with us and with biodiversity conserved. And I just wanted to thank my colleagues at the Complex System Lab. Some of them helped me to teach this course over the years. And I think that would be all. Thank you.
Thank you very much, Ricard, for this great and very inspirative talk. So we're in the midst of an emergent system, which means that we are late. And I guess we'll squeeze a bit the coffee break and give just room for the discussion as if we were not late. So, any question in the room? Yeah, maybe because there are people on Zoom, so if you can use the mic so that they can also listen to the question.
No, nothing. Now? Yeah. Yeah. Very nice talk for a Monday morning.
Thank you.
Say that. Nice way to start the week. So I had a question about the slide that you have. Do you remember that you have the rate of mutations and the size of the DNA of an organism?
Yes. Mm-hmm.
It was a very interesting
Yes,
Right.
which actually includes not only viruses, but...
So I want to ask, how did they define the genome size before getting the results? Like, what was it, like, how did they define a genome there? Like,
Well, no, the prediction from Eigen and Schuster was done at the time, but even genome size was not very well known, right? There was estimates of pair bases, etc., but not mutation rate or the number of genes or things like that. This is much afterwards. This is more recent stuff, of course, right? So there are estimates of genome size that are accurate, and the most difficult part of the story, mutation rates. How do you actually measure the effective mutation rate? So that fits with the prediction, but at the time they were doing that, they knew almost nothing, which is not bad, I think.
thank you.
I think there was another question over there.
First, thank you a lot for the presentation. It was really interesting.
Thank you.
My question is about the end of the presentation about terraforming and like the work and die slides. I was under the impression that like basically you were talking about gene manipulation in order to create a new entity that would be able to help us like save the planet, right? More
yeah, that's a bit brief way of saying that. But
more.
okay, let's say that. Yeah.
But my question is, like, I was under the impression that like gene manipulation is not really predictable, and you would just like have to see the outcome. But I understood from you is that you're talking about a way to not only evaluate the outcome but also control it beforehand, to interact with non-biological structures. I just want to, if you have time, like a small talk about this one, or if not, maybe we can have it in the coffee break.
Yeah. I mean, it's not a trivial question. I mean, there's that two answers if you want. The honest one is that anything that can be said about our proposal has to be tested, right? We'll see what happens. I mean, if we have learned something from biology is that there are no easy ways to solve problems, right? And a thing I want to point out is that we tend to think that if you deliver something, something is going to happen, probably not good. But the reality is that, and we know that from the few studies that happened in the 80s when people tried to do some little field work in that. There was a journal called Microbial Releases. I'm sure 100% that no one in this room knows about it. Nobody knows about it because it was essentially a journal that died in 2 years for failure, because essentially were very boring results. Like I deliver something, it stays there for a while, but nothing happens. It's not easy at all. But we have a lot of knowledge, and that's the second part of the thing. A knowledge from community ecology, and that's why the scales are so important. There's no much dialect between, much connection or communication between synthetic biologists working on the cell and the lab, and the people who knows about invasion ecology and community ecology. We know a lot about what kind of things that you can introduce in the system can be successful, and that happens only in some given circumstances. So that's why I think there's some predictability, right? And on the other hand, the idea of the manipulations we are suggesting, right, are on the scale of something that using other ways of working, right? For example, people has gone into the drylands and put cyanobacteria from the lab, right? And try to see if they help restoring the system. The evidence is that they can. The problem is that they survive only for a short while. So what about if we are more smart and we actually design, right? But not just being blind from the system's properties, right? Or the opposite. Let's go into what happens in the community, right? But again, we need to learn a lot on that.
Perfect. Thank you.
Thank you for the presentation. It was very interesting. My question is also about the terraformation. In the example of the concrete scratch, when it is all filled up, then when it goes out, the pH is different and it dies. But in in an open environment, how that stimuli of a stop would be applied?
Yes, that opens several different questions, right? One thing is that, as I was saying before, we might not necessarily want that this strain disappears. We might want to be there. And that's, again, something that is interesting from our understanding of invader species, right? We all have in mind the bad, really bad story, right? Like the rabbits in Australia, the species that for trivial reasons they are invaders. But there have been the vast majority of invaders, that would be kind of an invader, isn't it? I put something that is a bit different in an ecosystem. They keep thriving on kind of residual populations. So I don't have necessarily to want that. But if I have planned to have something that is going to be happy in this basic environment, anything outside, and the argument is a kind of a very, in the chemical level, it's really, really hostile, right? So you might survive, but you'll be just one more in the context of the ecology, right?
Thank you.
Thank you. Any more questions? So maybe I might have one, Ricard, maybe a bit naive, but in modeling, so you have different ways of building models. You have the data-driven models, you have the law-driven models, you have models that are more driven by conceptual thinking, such as those models that we've seen in theoretical physics in the last century, and you have models that are knowledge representations. So when you're developing the equations, for example, I've seen equations for gene stability. What kind of modeling approach are you using, and how do you handle the parameters and then the values of the parameters you need to feed? Okay.
Yes. Once again, it's as usual is what is the question you want to solve, right? Maybe you want to understand a given phenomenon, or you want to go beyond that and have more accurate predictions about clinical outcomes, for example. And that depends a lot on that. I can tell you, for example, what we are working now with a couple of colleagues, which is modeling, and it has a lot of tricky things computationally, modeling growth of a tumor where you have this instability at the chromosome level. It's tricky because every single cell has to contain a collection of chromosomes, which means strings of genes. You have to keep the information when you break down and you divide, and some of them go into another cell and the other cell. You have to keep the space under control. The model is hybrid and contains also a source of nutrient. So you have several things in place. What we don't have, for example, is mechanical forces, right? And I always struggle into in the future, what will be our picture of that? So how important will be one thing, one is the other. And I think that is an important thing also in terms of, again, communicating between people working on different areas. I have these colleagues working on physical models of tissues, really cool stuff, and you can go actually to the numbers. And they talk about cancer, but cancer is highly heterogeneous, and heterogeneity is not there, right? And probably to have both things is a very costly thing. What do we want to answer in this modeling? Our question is, can we give a good representation of how much instability at the chromosomal level is required to collapse the system? There's a number of things that are choices, right? Which is how exactly I do put, for example, parameters there, right? And that's something that we'll see. Our goal here is to see if, as it happened with the error catastrophe proposal that we made years ago, when you use different models, simple mathematical model with just cancer and normal tissue, then a heterogeneous population, then a computational model with space. If all the models are consistent in predicting that there has to be a threshold, you have answered a big question, right? There has to be a threshold. If you want to go a step beyond that and say, well, how, for example, a drug, which is something that we also are exploring, is going to help you in a solid tumor to actually go into the system and trigger that, that's another question, right? And which has more unknowns, and that's why you have to collaborate with the people in the clinics and in the drugs. That's why we're collaborating with people in the hospital. And as I was saying, I think that it's important to stress that. It's a relevant question to understand, and we want to, first of all, see if this is a positive question to that. A positive answer, sorry.
Yeah, thank you very much. I think you brought on the table a very important point, it's model consistency, actually.
Exactly.
That's something that very often in engineering we miss to do that exercise. Thank you very much, Ricard. So I think you deserve a good clap.
Coffee?
And a coffee.